A very good morning to one and all present here, and a special welcome to all the students and teachers for the second day of the event. Hope you had a wonderful time yesterday, and we hope and trust that you will have a pleasant experience today as well. We are extremely glad to have in our midst Dr. AnjuChadha from IIT Madras. Dr. AnjuChadha, a bioorganic chemist who completed her PhD from the Department of Organic Chemistry, Indian Institute of Science, Bangalore, India, in 1984. She received the Fogarty International Fellowship, USA, and worked in the Bioorganic Laboratory, National Institute of Digestive and Diseases of the Kidney, National Institute of Health, Maryland, USA, from 1985 to 1988. She then was a senior scientist at SPIC Pharma, R&D Center, India, and as an Alexander von Humboldt Fellow, she was in Germany from 1992 to 1993. She was a faculty member at the Department of Chemistry, Indian Institute of Technology, Madras, from 1998 to 2004. She then moved to the Department of Biotechnology and is a professor presently in that department. She is an awardee of International Women's Day from the University of Madras in the year 2011 for her contributions in the field of science. She is also a recipient of Professor S.K. Chatterjee Outstanding Women Researcher Award from IISc Bangalore in 2016. Over the years, she has mentored several PhD students who are well placed in academics and industry. She continues to carry out research with students and postdoctoral fellows at IIT Madras. Her research interests include biocatalysis and enzyme mechanisms, enzymes in organic and asymmetric synthesis, green chemistry, biosensors, and more recently, the chemistry of renewable resources. Over to Dr. AnjuChadha.
Some of you. Yes? How many of you are from IIT Madras? KV? I think we've met before. So you'll excuse me if some of the things are familiar to you in this lecture. And it's really a pleasure to see so many bright young faces. Always delights me, which is why I'm here. And I'm sure I'll try to make an effort to keep you from sleeping on this nice rainy morning. Yeah? So without much ado, this is going to be a lot of interactive kind of talk, yeah, because you know, people know I'm in the habit of asking questions as I go along, so you must pay attention and ask questions. That is the first thing about science. No matter where you are, which field you are, which milieu you are, which frame of mind you are, in whichever setting you are in life, ask questions. Question everything. Question tradition, question custom, question phenomena, question natural phenomena, question unnatural phenomena. It's about questions. What's the meaning of Jigyasa? First slide. Curiosity. And do you know why this series of this program? What are we here for? Just another lecture. What are we here for? Yes? Any thoughts? Yes?
Spread the curiosity among the students.
To spread curiosity among the students? Can you actually do that? Yeah, but you're pretty close, right? So we would like to. Why is that as we go along, right? Why do we need more science in this country? Do we need more science in this country? How many of you are convinced we do? Put up your hand. No, you don't need science in this country?
Yes, we do.
Yes? But science and rationality, I'm changing these words, interchangeable words. Do you or do you not need it? We need it. I hope you're convinced, which is why you're here. Right? And you need to ask the right kind of questions and persistently, not get carried away by fashionable trends. You like a problem, you want to figure it out, go at it. It doesn't matter what the world thinks. At this point, I want to tell you a few sentences about the discovery of this drug called artemisinin. How many of you know that artemisinin is the latest in anti-malarials? And a couple of years ago, a Chinese lady, Yu too, got the Nobel Prize for this. And do you know how it was discovered? She didn't care what the world said. She was at it with a team in China for 45 long years, taking clues from Chinese traditional medicine and the political situation then in China, Chairman Mao and the works, don't worry about all that, but she kept to it. And she had a small problem, it was not soluble. She changed a small derivative and made it soluble. And it became the drug of promise for malaria. Of course, mosquitoes are smarter than us or smarter than we think. So you're even having problems with resistance to malaria right now, to artemisinin. But because of her absolutely devoted work, we got this beautiful natural product called artemisinin, which is a drug. You know, after she got the Nobel Prize, the reporters went to her house, a small, modest house, and all she said was, I have nothing more than all my data and records over here to show you. That was her prized possession. That kind of madness for work, for pursuing a problem. So it doesn't stop with just curiosity or intelligence, it has to be backed with a lot of hard work. Also, if you're an experimentalist, be prepared for long hours of hard work and being alone. It's not about chatting with friends. It's about getting into yourself and thinking about your problem. Soaking yourself, saying, I will solve it. Okay? So this is where we are at with respect to what we think of research. And it is a lonely existence. Believe me, it's not easy. It's not for money. But the high you get when you have solved one problem, the high, the good feeling, and how it opens out a lot more questions to you, that is what you should get addicted to. Then you will see the fun in science. Alright? But this is all above and beyond, and I will just tell you a little bit about some general considerations in life sciences with biomacromolecules and what all could have gone into this hard work to get this far is what I want you to contemplate. I want you to go away with this feeling that this must have been tremendous amount of work and there's still a lot more to do. Okay? Alright. So, it's about chemicals of life, and since I work in the area of enzymes and organic chemistry's perspective to mechanisms in natural phenomena, I'll concentrate on biomacromolecules. And I'm not sure you can see my friend over here, the black buck. People who haven't been to IIT should do that just to pay tribute to this endangered species. We are very proud of this. Alright. And you know, the nice thing is you must notice how these pair of antlers have a symmetry about them. One is right-handed helix, the other is a mirror image left-handed helix. So chirality is very important in nature. What is chirality? What is chirality? Kairos meaning handedness. Look at your hands. All of you look at your hands. Put one on the other. Are they superimposable? Are they superimposable? No. So, if you have molecules or have objects, mirror images of which are non-superimposable, that is the phenomena of chirality. And all molecules in nature, you will see are generally chiral. In fact, if you go to a different planet and you're asked if there is life and you find a chiral molecule, you can almost be 100% sure that you would have life if the molecule was chiral. Okay? All right. So, this is about inspiration from nature. Everything in nature will inspire you. Believe me, everything. All right. So, this is my favorite cartoon character. Who's he? Kelvin. How many of you like him? Only few people. Oh, better get to read Kelvin. He really puts you in a good mood, doesn't he? So, he says curiosity is the essence of the scientific mind. Actually, it's a bigger comic strip. I didn't bring it because I thought it would offend the boys over here. So, I let that pass. Yeah? So, this is showing you that no matter what life system you're considering, whether you're considering unicellular, multicellular, plants, animals, there are many biochemical processes which are the same in all organisms. Doesn't that make you wonder? How would this design have come about? Why is it that everybody who needs energy must go through a specific pathway with minor deviations, minor developments, and what we perceive of as evolution, a step, what we say, ahead. But generally, I am one of those who always wonders why we say that multicellular organisms or complex organisms are more evolved, when actually a unicellular organism can do practically everything that a multicellular one can, except probably talk and few others. So, now I really don't know who is more evolved. Nevertheless, the biochemical processes are pretty much similar. So, if you make that as a starting point, then you really wonder, if you understand one, can you extrapolate to the others? And why is that important? Why do we want to know? Coupled with this curiosity should be also a use, an application. An understanding always empowers you. If you've understood it, there can be some control, there can be some inhibition, there can be some enhancement, especially when there are diseases, especially when you want to improve certain life parameters, right? Yes? You with me? You looking through me? No? All right. So, if you look at the biochemical processes, the smallest parts of those organisms are molecules. You know, science is very difficult. These processes are very complex. So, the business of science has always reduced the problem to making it simple so that we can tackle it, deductive logic. There is a flip side to this. Way back, Schrödinger had written a book on what is life. Those days, people wanted to look at it as a complete phenomena. Today, we realize that if we look at these phenomena in isolation, we may have understood a little bit more, but the thing to do is to put it all back together and understand an entire system. So, it's given rise to a new discipline called systems biology. If you tamper with one component of a cycle, you can see that you will have changed something else in the cell, overall composition, overall movement or kinetics. So, the idea is to understand the whole process, and that's important. Yeah? Look at this. The biochemical cycles repeat over and over and allow living creatures to survive. What is the ultimate reaction for life? Sunlight producing food or sugar with the help of carbon dioxide and photosynthesis, right? Have we been able to replicate that in vitro? No, not yet. We are trying. It is such a fantastic system, and that gives rise to this particular cycle but then all food gets again recycled as glucose. Again, the cycle repeats back to the earth, back to plants, back to food, and it goes on. Little surprise, it's called a cycle. It's a cycle. So there is repeatability. This is what biochemical pathways in a cell when mapped can look like at this point. It could be even more complicated. And if you think you can't see this clearly, and if you think this is absolutely intimidating and you get scared and what the hell this is, that is precisely why it's there. You're supposed to react like that, because I'm emphasizing that this is so complicated. And you can see that one would have just magnified a small portion of this and understood the biochemistry or the chemistry or the biology. Then you must relate it to most things over here. Can you now imagine how difficult it must be for medicine to appreciate what is going on in the human body? Yeah? Doctors are not gods. They don't know the answers to every problem. We don't understand as scientists everything in a cell. Okay? So this is how complicated it can look. Now, furthermore, if I just simplify it a little bit, you can see that if you have proteins, carbohydrates, all this in red is proteins, and somewhere it connects with this cycle in blue, somewhere it connects with yellow, with lipids, it also connects with carbohydrates, and it gives you energy over here. So it's all connected. Yeah? Every cycle has a place, and each one is just a small piece which needs to be fitted in the rest of the mechanism. Molecules are used as reactants and transformed into products. Life is one big network of activity where each piece relies on all of the others. Yeah, you cannot really ignore any part of the body, in fact. Even your nail, even the hair. Everything is ultimately connected. So you have this cycle, which is very well known, the Krebs citric acid cycle. You have studied this. How many of you have studied this in school? Yeah, it's a very important and basic cycle, yes? So you have 10 steps in this. But I'm not going to talk about cycles. You need some water? We have these kinds of cycles, plenty. And most people, most students whom I teach tell me biochemistry is very boring because we have to remember too many cycles. You don't have to. Who told you you have to remember it? You have to understand it. You go one reaction at a time and you see, okay, now I start with 6 carbons. This is breaking up into 2, 3 carbon units. How is that happening? Related to some organic chemistry, and believe me, it becomes more interesting. So if you come from a chemistry background, I believe that it helps understand better. Yeah? So we have to simplify. Very complex, very, very complex. Let's simplify. 2 main chemical processes. Which are they? Yes, which are the 2 main chemical processes? Read it out, you can see. Loud. You had breakfast. Loud.
First is photosynthesis, second is glycolysis.
And what is photosynthesis? What does it generate? Glucose. And what does glycolysis do? It breaks down, right? Please sit down, thank you. So you have one which generates glucose, the other which breaks down glucose. And that's enough. Yeah? But then what is glucose? What is glucose? 6 carbon compound. Anybody wants to add to that definition? Benzene is also 6 carbon. Benzene is glucose? Yes? Who wants to say what is glucose? Anything about glucose? It's a carbohydrate. It's a carbohydrate. What do you understand by the word carbohydrate? Carbohydrate. It's a hydrated carbon, which means it must have water, H2O, with the carbon. So if I write CH2O bracket, I can get glucose. Yes? So we can see that this is so important. And why are carbohydrates important? What do? They give us energy. They give us energy. And that can happen with the breakdown by this process. Correct? Now it's not as simple as having only glucose, because there are variations of carbohydrates, right? Sugars also fall into the same category. So this is the first class of biomacromolecules which are exceedingly important. You have nucleic acids, lipids and proteins, which are the others. So in addition to carbon, hydrogen, oxygen, you must also have nitrogen somewhere. A very complicated way to have nitrogen in the body. Yes? Carbohydrates don't contain nitrogen, but which molecules contain nitrogen? Proteins contain nitrogen, and amino acids are proteins. Yes? What other class? Big class? Nucleic acids. Right? So there are two classes mainly which will contain the nitrogen, two which will not. Which is the other one which doesn't? Largely. Some of it does. Yes? Lipids. That also. Sphingolipids can contain nitrogen. Also some aza sugars can be carbohydrates. So nitrogen is another element which we need in plenty. Okay? And which are the four classes again? Nucleic acids, carbohydrates, lipids and proteins. Okay? Here you go. You have carbohydrates, lipids, proteins and nucleic acids. And all these put together give you the life processes. Are there anything else? Yes. Other molecules? Yes. There are secondary metabolites in plants. What are those? You know an example? An example of a secondary metabolite, which may not be necessarily this. Some medicines, I just told you about artemisinin, right? Artemisinin, alkaloids, anthocyanins, pigments. You see such beautiful colors if you ever go to a vegetable or a fruit market. All those are also part of the chemistry of life. What else? What about minerals? What about minerals? We also have plenty of those. Yes? Any mineral you can think of which we have, which we need absolutely? Calcium, very good. Potassium, sodium, selenium and so on and so forth. Alright? Now of course, so you break down all these big molecules, macromolecules, to smaller units, and you will see that ultimately you are talking about carbon, oxygen and hydrogen, the atomic structure of which is known, and if you understand that, you can get a variety of your understanding of various structures that are possible. Alright? Here you have molecular weight less than 1000 micromolecules, greater than 1000 macromolecules, which are biomacromolecules. They are generally huge chains of monomers present in the body. If you were to generate these polymers in vitro in the lab, it requires huge amount of thinking, energy, protection, deprotection, but nature is doing it all the time. All the time, As you sit, as you listen, as I stand, as I talk, we are breathing, digesting, circulation, nerve impulse, and the works. It's all happening simultaneously. And who's catalyzing this? Molecules which are known as enzymes. They know their job. They're not getting into each other's way. And they're still happening. Can you believe how marvelous this design and this orchestration must be? Yeah? All right. So we have here carbohydrates. We've already seen that. In addition to glucose, you have fructose, maltose, sucrose. And glucose and fructose in fruits, sucrose is stable sugar, maltose is germinating barley, and you have C12H22O11. The same structural formula but different, same molecular formula, different structures. What are these called? Isomers. Great. So you have lot of complications in structure. You know that. For example, we can have starch. Can we digest cellulose? Can we go out grazing with the cows? Can we? You know why? Why cannot we eat cellulose? Yes, yes, who wants to answer? Just put up your hand. We can't hear otherwise.
We do not have the cellulose digesting enzymes.
What is it called?
Cellulase.
Very good. We do not have the enzyme which can chew up your cellulose. And what should be so difficult about an enzyme which can do the same with starch, but it cannot do with cellulose? Small difference in structure of starch with cellulose. So enzymes are so specific that they are looking at every detail of structure. They are even looking at three-dimensional structure, and if something is above or below the plane, that's a difference. That's how specific they are. Again, it's absolutely mind-blowing that they are so stereospecific. Yes, stereospecific. All right. So you have simple carbohydrate sugars, glucose, you have them in grapes, honey, grains, etc., etc. And this is the structure of glucose. A pyranose ring, there is an oxygen, 6-membered ring, you have CH2OH, OH, 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 OH. This is the position which we are talking about, whether these OHs are above or below the plane. All right, this is important to remember. Other carbohydrates, you have maltose, you have sucrose, and you have these polymers, starch and cellulose, right? You have sucrose, which is a disaccharide. Maltose, another disaccharide. But two identical glucose molecules. Here you have glucose and fructose. What is the difference between them? You have the CH2OH here, OH here, OH here. Look at the difference. Yes, the positioning of the OHs. And this joining bond is the glycosidic bond. Important to remember. You know, if you have a problem trying to memorize biochemistry, try this. You just get to the structure, understand that, and things will be easier for you. And they are quite fascinating. You learn to love to draw them also, if you have that small inkling to draw. Then you have starch. Look at the difference between starch and cellulose. We made a case. This is all CH2, CH2OH, but you have one CH2OH here, plus this is down, this is up. So there are differences in structure. There is also branching. So all that adds to different structures between the two, which the enzyme sees. That is amylase versus cellulase. So we can't eat grass. But have you seen a cow eat bread? Roti? Yes. What does that mean? What does that mean? Cow can digest the roti. Starch probably. What does that mean? Who's more evolved? Don't have to answer. Okay? So you can see that there are very many such examples in nature. All right. Now, I just told you, you have maltose with 2 molecules of glucose, and fructose and glucose come together to give you sucrose. You have cellulose again. This is what we call a beta 1,4 link, beta above, alpha below, and cotton fibers. That is the purest form of cellulose. A little aside here, these days people are working a lot on renewable resources, and there is wonderful work going on with cellulose. People are actually saying, September I was in Russia giving a talk, and there in international forum there were people telling you that silicon will be replaced by cellulose in a lot of applications. So there is a lot of good chemistry happening with cellulose and lignin. Okay. This is starch. This is alpha 1,4 or 1,6. I told you it could be branched, and you have variations in the structure. You can have helical or amylopectin, and you can see that enzymes which hydrolyze these. This is what it looks like, granules of wheat starch stained with iodine. You can see the iodine color with starch. It's a tissue. Complete that. We have to learn to do this. We have to learn to define properly. Only then we understand. The question was, what is a gland? What should be your answer? Complete answer. Okay, we'll agree with that? We'll give a 10 on 10 with that? No? Yes? You won't? Did you say that? So, what is the lesson? That we learn to be a little more specific. We learn to think a little harder. Also, language has to be precise. Science is about precision. You can't, it's not about, this and that and storytelling. Okay? You have lipids, oils and fats as the next class. Which oil do we use in Madras generally for cooking? Sunflower oil. Why? What is local? What is local to this state, this country? South India. Gingerly and mustard. These are our local oils. Where the hell did sunflower come from? Is that local for us? Why are we using it? Ever wonder? Ever wonder? Why? And why is it easily available? And why is that happening? Profit making. And who's doing that? Where does it come from? The MNCs. India is the market for all this, right? Because we stopped thinking long ago about for ourselves. We stopped thinking that our history, our geography is more important. Our local produce is more important. Our bodies actually tune very well to our atmosphere. So why shouldn't we be using our own oils? Yeah, but my problem is what happened to that brain? Why did it get washed? Okay? So think about these things, and all lipids, even the extraction process has to be questioned. So you just keep asking why, and you will reach a place where you will begin to understand. That's what I just did, right? You have a glycerol molecule. What is glycerol? It's the triol. It's the simplest sugar, so to speak. OH. CH2OH, CH2OH in the center, CHOH. So there are two primary alcohols and one secondary alcohol in the center. All right. So you have OH, OH and OH, and these alcohol groups can be esterified by fatty acids. Fatty acid, any acid plus alcohol gives you ester, right? So if you have glycerol and you have 3 molecules of a fatty acid, you get glycerol which is esterified in all the 3 positions. This is a triglyceride, and most fats and oils are triglycerides. Those fatty acids differ from crop to crop. We said coconut oil, we said groundnut oil, we said mustard oil, we said gingerly oil. For your own reference, you should write these 4 oils, find out what is the composition of these fatty acids in these oils as opposed to sunflower oil. You will understand why it's important. Largely, you have saturated or unsaturated fatty acids. You can have stearic acid, oleic acid, linoleic acid. Yes, any of these fatty acids. Triolin is, or there's another craze to use olive oil in India. This is bizarre, according to me. The Mediterranean can use as much olive oil as they want. We can use it for flavors. I'm not saying don't use it, but the way it is being marketed is ridiculous. You think that if we tell them buy gingerly oil or buy groundnut oil because it's good for you, you won't break ice with them, right? So one has to also consider few factors of physiology, local geography when it comes to these things. Triolin will have oleic acid in the 3 positions. That is why it's called olive oil. Stearic acid has unsaturation, it solidifies. These days there is a big craze to eat a lot of coconut oil. I am sure you read about it. Thanks to social media. They show you clips where Americans get up in the morning, take out from the fridge one jar of frozen almost coconut oil and take scoops of it and eat it, because they have been told that coconut oil prevents dementia. It's helpful for Parkinson's and Alzheimer's and all. It contains lauric acid, fatty acid. It's good for you. Yeah, but that doesn't mean that you go crazy and start having kilos of it. Okay, so these are some of the things which we know associate with fatty acids. This is coconut oil, butter, whole lot of triglyceride. Here, this is what I have shown, what I was just talking about. Yes, and you have, you can generate this in the lab. You can actually make these triglycerides. You can even hydrolyze these triglycerides, and we do that all the time when we have projects to do this. So there is also the whole aspect of technology which backs up science research. There was a time when it was very difficult to analyze lipids. Today it's very simple. You can actually have chromatographic methods which can tell you which triglyceride has which fatty acid. We do that. Okay. So this is it. He's saying, hey oil, you want to hang out? He says, I can't mix with you guys. They call him a hydrophobe. What is hydrophobicity? Who hasn't answered? One of the guys. They'll all think that I'm very partial to my girls. Yes? What's hydrophobicity? The molecule which repels water. Hydrophobe. What does that word mean to you? Good. So if you understand the word, you know the definition. And why does this happen? No, why does this hydrophobicity happen? Why would some molecules be hydrophobic and the others be hydrophilic? Okay, let's look at what is hydrophilic. If you know an example of what is hydrophilic, you will understand what is hydrophobic. Give me an example of hydrophilic molecule. Alcohol, okay, good. So, what happens in an alcohol? But I can't hear everybody together. Somebody? Yeah? I'm asking why. What is so special about that structure as opposed to oil, as opposed to triglyceride? This also has, triglyceride also has, no? O's, it has O's. It's an ester. Okay, do salts dissolve in water? What is clear to you about salts? No, property. Huh? Sorry? Yes, they are charged, ionic. Now take a clue from that and tell me why alcohols will dissolve. Polar. You got to understand polarity of bonds. What does that mean? What happens in OH? Delta positive, delta negative. What happens in oil? Very less charge separation. Lot more of carbon hydrogen bonding. Okay? There is nothing like even remotely making it ionic or charge separation, which makes it hydrophobic. Yes? Anything else which you can think of which is hydrophobic other than oil? Any organic solvent which doesn't go into water? Yes, yes. Mercury, is that a solvent? It's a liquid metal, it's a special case. Yeah? Yes? Kerosene, okay, hydrocarbon, many hydrocarbon solvents, chloroform. What happens if you take chloroform and water in a beaker? Who will be down, who will be on top? Chloroform will be? How many of you say chloroform will be on top? If I take oil and water, who will be on top? Oil. Clearly hydrophobic, floating on top, and water down. I have taken chloroform instead of oil. What happens? It also doesn't mix. Does it come on top?
Yes, ma'am.
No, it doesn't. So it's not always that it's lighter. Somebody talked of density. That's not what it is. Please go. How many of you have access to a lab? How many of you have access to a lab in school? A chemical lab? Everybody. Please do this experiment. I'm sure you will have chloroform. Put it in a test tube with water and see who goes where. Alright? Don't inhale too much chloroform. Why? What is chloroform used for? Huh? It was a very, very common anesthetic molecule. So if you keep inhaling a lot of chloroform, you might swoon. Don't do that. Just do this cute experiment. Okay. Watson and Crick. And again, it was to do with a lot of wonderful stories. As always, the women in the game got the raw deal. Rosalind Franklin did the major work, in fact the key work, and didn't get any recognition for it because these two guys grabbed all the attention and so on and so forth. Read about it. Very good history. And you have nucleic acid structure which gave a real boom to molecular biology, diagnostics, understanding genetics, and so on and so forth. So you have the structure, and you have, incidentally, nucleic acids are made of sugar phosphate and a nitrogen base. Why are they called acids? if they are made of nitrogen bases, why should they be called acids? Who's they? Where is the negative charge? Yes. Who wants to answer? Yes. You have a nice badge. What does that say? What does that say? House captain. Okay, house captain. Why is the nucleic acid an acid? Don't know. What it has a base. It has what side chain? No. No. Nucleic acid structure is covered in your portions or not? Yes. So you have a phosphate, sugar phosphate diester on the sides. Now, why is it an acid? It's not a protein, it's not a nucleic acid. Find out. Find out. There's a phosphate somewhere. He said something about a negative charge. You should know that. Sugar, phosphate, and nitrogen base. There's an OH, right? The phosphate group. That is why it's an acid. He said that OH can go, you get the O minus. That is why they are all acids. And you have many applications. You see in forensics, which a lot of young people are interested in for some unearthly reason, which I find terribly morbid. And you have a whole lot of genetics and who were your ancestors and so forth happening with DNA sequencing, identification and all that. This is what I was talking about when I said minerals. You see how many major elements and how many trace elements we have in the body. Right? You have lot of water in the cytoplasm. How much of the cell is made up of water? You are how much water? 70%. So there is lot of water. In addition, you have the biomacromolecules, you have minerals. Calcium, phosphorus, potassium, sodium, chlorine, sulfur, magnesium. Iron, zinc, copper, molybdenum, selenium, iodine, manganese, cobalt. Right? So huge amount of chemistry going on, both inorganic and organic. Salts of sodium and potassium, very, very important in which system? Which system of the body?
Pumping of blood. Pumping of blood. Blood.
Blood, yes, nervous system. Very, very important. All impulses being controlled by flow in and out of all these ions. And what are those? The mechanism by which these ions can go in and out of the cell? There are channels, ionophores. Few years back, this work got the Nobel Prize. You have to wonder about it. It's very easy to say it goes in and out of the cell. How? Where? What governs it? What regulates it? What can go wrong with it? How can we correct it? Right? These are the questions. Proteins, my favorite because we work on them. Also because I think they taste very good. Certainly the egg does. How many of you fried an egg? Seen a raw egg and fried an egg? Yeah, put up your hand. Yes, tell us what changes you see. Who wants to tell us? Somebody from there? Yes? What does a raw egg look like and what happens when you fry it? Yes, it's a raw egg has transparent kind of jelly-like stuff, and you put it on the pan which is heated. What happens? It turns. Who turns white? There are two portions, no? The transparent portion becomes opaque. It becomes white, and the yellow becomes. It remains yellow, but it looks bright. Gets broken. What is a hydrogen bond? asking the? Talking of proteins. We're talking of proteins. Bases come where?
Substrate when enzyme is added, the process is going fast, and in the end of 20 minutes the work is done.
The starch is formed. Good. B.
B, substrate water is added but no reaction is taking place.
Okay. C.
There also no reaction is taking place.
What have you added?
Enzyme and water.
Therefore, what is your conclusion?
So that will be enzyme substrate.
What is your conclusion?
Enzyme is necessary for the process to happen.
With the substrate, because this also has an enzyme, right? So, conclusion is enzyme and substrate together are giving us the starch. Why are we doing B and C? Sit down. Very good. Who wants to answer why are we doing B and C? What does that tell us? These kinds of experiments, B and C, called control experiments. Control experiments, very important. Yes, substrate plus water or enzyme plus water doesn't do the job. Therefore, we definitely need enzyme plus substrate. Can you avoid doing B and C? No. That is what science tells you. You must have proper controls for every experiment to make a reasonable, logical result. You can't get up and tell me, I just added this to this and I saw starch, therefore this is important. No, I had my substrate in a liquid. There was water. Was water doing the job? That will be the most obvious question. So you must avoid all this ahead by doing proper control experiments. Got it? Very good. This wonderful molecules for us, pushing the boundaries of their substrate scope. That is a green approach because they are biodegradable. No need to add a mess to the already messy environment, right? Okay, so this is the last slide. I think I'm on. I hope you enjoyed it as much as I did. Any questions I will take.
Ma'am has just said that in her laboratory they extract enzymes and then they use it to make other biomolecules. I wonder, how do they extract enzymes from the biomolecules, ma'am?
Okay, so good question. Also, let me just tell you that there is a whole I have the mic. There is a whole discipline for protein purification. So there are protocols which tell you how to purify from whichever tissue or microbial cell that you're starting with. It's not always necessary to purify an enzyme. We are using whole yeast cells also to do our reactions. So that's a perfectly doable thing. Also, I will warn you, it requires tremendous amount of patience. But when it works, it's almost like magic. When you see a single band on the gel, which is a picture which shows you how pure it is, when you started, you had some countless bands, and you have gone through the protocol to get a single band or a single crystal. That is joy undefined. I cannot even explain to you. Okay? So there is an experimental methodology available. Yeah.
Protein supplement.
Yeah.
It is benefit for health or not?
Oh, there is no one universal law of that kind. It depends on where and who is deficient and whether you need the supplement. It's a good idea to avoid supplements and just stick to natural food. Your natural activities and food. So there's no need to overburden your system with synthetic stuff and supplements, in my view. Yeah.
Thank you.
Ma'am, you have kindled all the young minds and made them more curious and inquisitive. We have a small token of our appreciation here. I now request our coordinator, Dr. Tamil Selvi, who is the principal scientist in CSIR-CLRI to hand over the moment.